Good evening, everyone, and thank you for joining us this evening and for choosing science over the pub. My name is Nikita, and I'm a postdoc at the University of Oxford and a Pint of Science volunteer, and I'll be your host this evening. We have an awesome selection of guests this evening for Beautiful Mind, here or here. Before we get started, please do tweet throughout the event and use the hashtag Pint21. We usually get this trending every year, and we want to keep it trending again. And so I'll just give you a quick run through. First, we'll have our first speaker, Kerry Walker, present her research, followed by a Q&A. We then have a fun quiz lined up designed by one of our talented volunteers. And after that, we'll hear Armin Lak discuss his research with some questions, and then we'll present the results of the quiz. Now, I'll stop talking because you didn't come here to see me, and introduce Dr. Kerry Walker, Associate Professor of Neuroscience from DPAG, Department of Physiology, Anatomy and Genetics, University of Oxford, in auditory neuroscience, who will explain how our brain makes sense of complex auditory sounds. Thank you, Kerry.
Okay, thank you very much, Nikita. And thank you for the invitation to speak today. I hope this is of interest to a lot of you out there. So I'm going to talk today about hearing with your brain, which is what I study here in the Auditory Neuroscience Lab at Oxford. So hearing with your brain, where do we start? Well, a good place seems to be to start with a sound source. So objects in our environment can vibrate, like piano strings, for example. And when they vibrate, they produce sound waves. So these are changes in the energy in the sound, in the air rather. Sound pressure waves enter our ear canal. They start a motion in the tympanic membrane, known as the eardrum, probably to most of you. So this back and forth movement of your eardrum changes in amplitude and in frequency as determined by the waveform in the sound. Now you notice this sort of snail-shaped creature at the back of your inner ear. This is known as the cochlea. And this cochlea has the job of changing the movement of your eardrum into nerve impulses. These nerve impulses travel through lots of different stations in your brain that are a part of the auditory system, and ultimately they make their way all the way up to an area called the auditory cortex. Now, although there's a lot of brain areas involved in processing sound, and they're all very interesting and important, I'm going to focus today on the auditory cortex. And this is of particular interest to me because we believe that the auditory cortex is the seat of our conscious perception of sound. And what I find amazing is that the millions of neurons in our auditory cortex can underlie our representation of sound itself. So they change this simple back and forth motion of the eardrum into our mental imagery, our experience of melodies, a familiar voice, and what have you. We have a very rich inner world. And so how on earth do our brain cells in auditory cortex allow us to take a sound wave and turn it into this very behaviorally useful mental world? Well, that's a very big question. So I'm going to narrow it down for us a little bit today into a slightly more tractable question. And we'll focus on how our brain allows us to perceive the pitch of sounds. So what is pitch? Well, pitch is our perception of the tonal quality of the sounds that we hear. It forms the basis for our perception of melody in music, but it also plays an important role in our perception of all the sounds that we often find in our environment. So it allows us to communicate. You know that as you speak, you change the intonation or the pitch of your voice. Babies are attracted to high-pitched voices because they sound more like mothers. And animals also use pitch cues in their communication. They might make low-pitch voices if they want to convince you that they're big and scary, for example. Okay, so let's start with the ear. So how are sounds encoded in the ear? Let's go back to this snail-shaped structure called the cochlea, and let's see what's happening inside the cochlea. But down in this little video, I'm going to unwind this snail-like creature so that we can better visualize this membrane inside it that's called the basilar membrane. There it is. And now let's see what happens when we play some very simple sounds to the cochlea. So when we play sounds, the basilar membrane starts to vibrate. And when the frequency of the sound changes, the basilar membrane vibrates in a different region. That happens because if we look at the cartoon of the basilar membrane up here, the basal end is very stiff and narrow. And so that likes to vibrate at high frequencies. And then this resonant property of the basilar membrane changes all along its length to the very wide and floppy end that likes to vibrate at very low frequencies. So along the basilar membrane, we have hair cells that are sensitive to its local vibration. And they pass on these messages to the first neurons in the auditory system, the auditory nerve fibers. And so in this way, they set up like a map of frequency where neurons in this end will respond only to high frequency sounds and in the opposite end to low frequency sounds. And this neural map of frequency that we call the tonotopic map exists from the very earliest stages of the auditory system and is preserved through most stations all the way up to even the auditory cortex. So, is that our question answered? I mean, that would be a pretty short Pint of Science talk. Are we done here? So we have low piano keys on this end in these neurons and high piano keys in these neurons. Well, I'm afraid it's not that simple. So most natural sounds, unlike these pure tones that I was just playing, contain many frequencies. And that's shown here. So this is a spectrogram, and the color scale shows you how much energy is present at different frequencies going from low to high across time. So this is just a spectrogram of a piano chord. It's just a middle C. And you see you've got these bands of energy all across the frequency range. So actually just one piano key contains loads of different frequencies, even though we hear it as having one pitch. So now if we play music to the basilar membrane, it's going to get a lot more complicated. Let's try that again. So now we get energy across the entire membrane, and we can't use this simple place code to figure out what pitch we're hearing. Well, here's the brain again, stepping into the rescue. And scientists that have studied the auditory cortex, and particularly looking at what happens when we have damage to our auditory cortex, they notice that people with this type of neurological impairment can still discriminate two pure tones of different frequencies pretty well. But if they're asked to make judgments of musical melody or to discriminate the pitch of more complex sounds, then they show impairments. So it seems that the auditory cortex is somehow allowing us to hear a single pitch of this really complex frequency spectrum here. So we're going to try and figure out today how that happens. And I think we'll have to start first by looking at what is it in a sound that makes it have a pitch. If it's not frequency, what is it that the brain is looking for? Well, there's two things. So what I'm showing you here are two different sounds, a piano note and a voice. And I hope you notice that although these sounds sound different, one sounds like a piano, one sounds like a person, they share something. They share a perceptual quality we call the pitch. So they're both at middle C. Why do they both evoke the same pitch percept? Well, acoustically, if we look at their spectrograms again up here, you notice that the voice has bands of energy, just like the piano note did. And it turns out that these bands of energy are all multiples of a common frequency we call the fundamental frequency. And in this case, it's 262 hertz. And lo and behold, we hear the pitch of these sounds at 262 hertz. So we think it's the relationships between these bands of energy that we call harmonics. These harmonic cues tell us which pitch we're listening to. But that's not enough. Well, it would be enough, but it turns out there's another cue. So we can ignore all this and look instead at these same sound signals, but in time. So here now, I'm just plotting the waveform over time. And you'll see that both of these sounds are repetitive. They have waveforms that repeat periodically throughout time. And how many times do they repeat per second? You can probably guess, 262 times a second. So the nervous system can either represent pitch as a pattern of activation across harmonics, or as a temporal code, a timing code that happens throughout the waveform and represents its periodicity. Now, just in case you're not convinced yet, I'll take it a step further and tell you that we can even perceive the pitch of a sound at 262 hertz if there's no energy whatsoever at that frequency. So that seems a bit wild. So this sound has no energy down here, but we still perceive the pitch down here. And although that sounds a bit weird and wonderful, you've probably experienced it most days of your life if you often talk on the telephone. So the telephone companies, to save a bit of money, they bandpass the sound signals that they send over their lines between about 300 and 3,500 hertz. Yet we can still hear perfectly the pitch of people's voices and the melody of music that's lower than that range. So that's just to drive home the point that the pitch we hear is not any one frequency in the sound. It's these much more complex harmonicity and temporal cues. So that's what we perceive, and it's easy to know what humans perceive because you can just ask them or you can listen to a sound yourself. But what about animals? Do they also perceive pitch in this weird and wonderful way, or do they just hear frequencies? Well, to get at this question, we designed an experiment in my lab where we trained ferrets to discriminate the pitch of sounds. So the ferrets would go into a box and they would hear some sounds through a loudspeaker at the front. And they heard two sounds every trial, and the sounds were either dropping in pitch or they were rising in pitch. And the ferret had to learn that when it heard a pitch decrease, to lick at the left spout and it gets a little water reward, or for a pitch increase, it licks at the right spout and gets a water reward. So can ferrets do this? Can they hear the pitch of complex sounds, regardless of what energy we put into them? Well, yeah, they can. So here's a trained ferret. I'm showing you now its performance. So I'm plotting here across the pitch of this target sound, the second sound, how many times the ferret goes to the right or the left spout. And the ferret correctly here has learned to go to the right spout when the pitch is high and to go to the left spout when the pitch is low. So it's not quite as good as our behavioral curves are on the same tasks as humans, but it does seem to certainly have a pitch percept. Now, if we go back to those harmonic cues and the temporal cues that I talked about earlier, we can manipulate our sounds in different ways to either remove the temporal cues or remove the harmonic cues. And in that way, we get at whether the ferret's brain is using both of these cues or one more than the other. And when we do that, we find that when the temporal cues are strong in our stimuli, the ferret performs very well. But when the sounds contain mostly just harmonic cues, the ferret's performance drops. So it seems a little more inclined to do pitch perception using temporal cues rather than harmonics. And this was surprising and interesting to us because humans show the opposite effect. So we train humans on the same task, and we're actually performing better with harmonic cues than temporal cues. So both ferrets and humans, and indeed in other labs, they've shown other species of animals can hear the pitch of sounds. They should have some experience of a change in musical melody, but we weight the cues, the acoustical cues, differently. Okay, now what about the brain? That's what we kind of wanted to start to understand, isn't it? So how can we record the activity of neurons in auditory cortex while we experience pitch? Well, something very useful about neurons is when they become active, they start shuffling these charged particles across their cell membrane. And so that means that you can actually record the activity of a neuron as a very tiny change in voltage just outside the neuron. And that's what I do. So here you see the voltage just outside of a neuron, and these spikes, as we call them, and you can see why, I hope, are basically saying the neuron had an event here. It had an action potential here, here, and here. And we just put a dot above each spike so that we can have a kind of trace of the output neuron. And you can actually send this voltage signal into a loudspeaker. And then you can hear those spiking events. They're really quite obvious, yeah? Okay. So now we're listening to a brain listening to sound. So it's all going a bit around and around here. Why do we do this? Well, when I'm listening to these spikes come in over the loudspeaker, it really sounds to me a bit like Morse code, yeah? If you kind of watch old war movies or whatnot, it sounds a bit like beep, 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 beep. And so now really our job is to decrypt this Morse code coming from this cell. So the cell is encoding something about its sensory environment in this pattern of spikes. And it's our job to figure out what that code is. But we've got a bit of an advantage over the people who did this in World War II, encoding codes from the enemy lines, because we get to choose what's being encoded. So that makes this decryption a lot easier. So here we have an experiment here where I'm showing you these spike trains on each horizontal line. And these are spike trains recorded from one neuron as I play sounds that are either very high in pitch or sounds that are very low in pitch. So the low ones here and the high ones here. And the neuron I've shown you here is using a very simple spike code, which is easy to decode because you can just see it when you look at this type of a graph of the spikes. So basically this neuron is sensitive to pitch, and it encodes it as how many spikes it elicits in response to the sound. So when the pitch is very low, it fires lots of spikes. When the pitch is very high, it doesn't fire as many spikes. So we find cells like this in auditory cortex, but not every cell in auditory cortex will encode the pitch of a sound. Some do and some don't. And some encode it in different ways than others using different codes. So now it's an obvious thing to ask, where are these cells that encode pitch? So this is a picture of the ferret auditory cortex, or the ferret brain rather, and in red I'm showing you the auditory cortex. And we'll zoom in on that here. So this is the ferret auditory cortex zoomed in. And in the color scale, I've colored neurons that encode a lot about pitch in yellow, and neurons that don't really care about pitch in the dark reds. And I guess the take home when I look at this is that you find neurons that are encoding pitch distributed all throughout the auditory cortex. So our hypothesis going in is, maybe there's one little pitch center or pitch region, but actually it wasn't quite as simple as all that. So we find pitch neurons all throughout the auditory cortex, and they're intermingled with neurons that are encoding other attributes of the sound, like where the sound is in space or what instrument is playing the sound. And in fact, many neurons, in fact, the majority of neurons here, encode more than one sound feature. They're not so specialized for one sound feature. Okay, so let's go back then to the behavior we found in the beginning. So we said that this is the plot from earlier. Ferrets perform better when I provide them with temporal pitch cues, and they perform not quite so well when I present them with harmonic pitch cues. So what about these cells in the auditory cortex? Well, if I plot now how many neurons, what percentage of neurons in the auditory cortex encode pitch when I present these different types of cues, we find that these neurons also seem to encode more pitch information when I present temporal information versus harmonic information. So that sort of fits with the behavior and tells us perhaps we're barking up the right tree here. But perhaps most intriguing is this third class of cells. So a small proportion of these neurons actually encode the pitch regardless of which type of cue carries the pitch. So these are interesting because these cells can tell you what pitch you're listening to regardless of the instrument that's playing it, or if it's based on high frequency cues or low frequency cues. So where are these very special cells then in the auditory cortex? And we find, well, these are very preliminary data, I should say, and we're still collecting data to answer this question. But so far, these more general pitch encoders seem to be more localized within the auditory cortex. So perhaps there's a bit of a more specialized pitch center here, which would fit with human studies that suggest that maybe there's a specialized pitch center in the human auditory cortex as well. Although I could go on all night about how contentious and unresolved that particular research question is. Okay, so if I have just 1 minute left, I'll tell you about one last experiment where we really wanted to address the question of whether the auditory cortex is really encoding our perceptual experience, our subjective experience of pitch, or is it just encoding the sort of bottom-up acoustical cues that are coming into the ear? So we have our ferrets doing the same behavioral tasks that I've already explained to you. And now this is the raster plot I showed you before, but we're going to record the neurons while the ferrets are performing the task. And what we can do then is feed the output of these auditory cortical neurons into our decoder that we've built on the computer and ask it one of two questions. So using the same neural spiking patterns, we can ask the decoder to tell us, did the ferret choose the left spout or the right spout on each trial? So this is essentially our behavioral readout of what pitch the ferret thought it heard. Or on the other hand, we can ask the same decoder with the same data, which sound did I present? So acoustically, did I present a low pitch sound to the ferret or a high pitch sound? And what we find is that if we now plot a choice index value, where higher values mean the neurons encode more about what the ferret reported about the pitch, and negative values would indicate what sound I played to the animal. Then at the beginning of the trial, when we first play the sound, the auditory cortex encodes both of these features. But as the trial goes on, it's biased more and more towards what the animal reports it heard. So this tells us that the auditory cortex kind of has a bias while you're listening to a sound to represent not what the actual pitch of the sound is, but what you perceived the pitch of the sound is. Okay, so that's all. I'm just going to summarize for you now what we went over. So how does our brain allow us to perceive pitch? Well, we compute pitch from the harmonic and temporal cues in a sound. Animals also experience pitch, but they depend more on temporal cues than we do. And pitch is represented by a subset of neurons in the auditory cortex. So I just want to thank many of the people who I've worked with over the years in Oxford, and the people highlighted in red, they're the ones that contributed directly to the studies I talked about today. So a great thanks to them, and a thanks to you for your attention. I'd be delighted to take your questions.
Thank you very much, Kerry. Thank you so much for an informative talk. And so now we have a couple of questions I'll pose to you from the audience. Question one is from Mike Richardson. And it's, in cases of tinnitus, aka the bane of his life, Is the basilar membrane actually vibrating, or is it a percept from other causes?
Right, that's an excellent question, and I'm so sorry to hear that, Mike. Tinnitus can be terribly debilitating and much more common than a lot of people realize. So there are lots of different causes of tinnitus. It's a very active area of research around the world, and particularly in the UK. It's probably not the result of a movement of the basilar membrane. So in many cases, tinnitus happens because those hair cells that I showed you in your cochlea have damage, and the auditory system tries to compensate for that by increasing their activation. And so what happens then is you hear a tone where there is no tone. But, you know, some people have tinnitus due to damage in the ear. Other people have damage, we think, due to damage higher in the auditory system, even as high as the auditory cortex. So yeah, unfortunately, a very poorly understood clinical condition.
Thank you. And then we'll go on to our next one, which is from Sapphira. Sorry if I'm pronouncing it incorrectly. Why did you choose ferrets as an animal model?
Okay, so can I screen share again, or is that too complicated?
No, I think we can do it.
Okay, because I often get this question. Thank you for this question, because I have a slide ready for it. Yeah, so ferrets are, of course, a great model to work with for many reasons, but they're particularly important for my studies of pitch perception. So what I'm showing you here in this graph is a plot of the audible frequencies. So what frequencies can different species hear? So if we look at, say, the mouse, which is a very common species in neuroscience research, they hear high frequencies really well. They hear much higher frequencies than we hear, but they're not very good at hearing the low frequencies that we hear. And because pitch is all sort of low frequency, that makes them a pretty poor animal model. So you want an animal model that likes to do behavior and can do lots of behavior, for which ferrets are renowned, and that kind of shares a lot of our audible frequency range. So I guess, you know, the cow would have been all right as well, but I don't want to try to fit cows in the lab, so I decided to focus on ferrets.
Brilliant. Thank you very much. And so for our next question, it is, so the first one is there variability between the amount of pitch sensitive areas in different animals? Is there a genetic component?
Okay, so I got a bit distracted because there was a different question on screen. Yeah. In the pitch areas was the question?
So the question is there variability between the amount of pitch sensitive areas in different animals? Is there a genetic component to this?
Yeah, absolutely, huge variance. So there's both differences in the sort of pitch threshold, so behaviorally how well animals hear pitch, as well to their weighting of these temporal and harmonic codes that I talked about. And in fact, the pitch area, which has been found in humans, has really only conclusively been demonstrated in marmosets, which is a species of primate. And so, yeah, certainly species differences, certainly genetic differences as well. But we hope in neuroscience, in the type of neuroscience I do, to find common principles that we believe will help us understand brains in general and not just your little subspecies.
Sure. I think we have time for one last question I've shown here from Sean. Can you explain a bit further the difference between a neuron encoding choice and encoding the actual pitch?
Right. So the neuron that encodes choice, well, when the animal is performing the task correctly, there's no difference. But the thing is, on some trials, the animal makes a mistake. Yeah. So the animal goes to the wrong spot. It was meant to go left and it was meant to go right. And then the question is, on that trial, did the neuron say that the pitch was correctly low, or did the neuron say that the pitch was correctly high? So on those error trials, the neuron will either reflect the ferret's choice or the pitch of the sound that we played, but it can't reflect both. So that's essentially what it boils down to.
Okay, brilliant. Thank you very much. With that, I'd like to thank Dr. Kerry Walker for her amazing talk. I know some of you have some extra questions. If we have time at the end, we'll go back to you. If not, maybe if Kerry can see the chat throughout, and maybe she could answer some of the questions during the next segment, if that's all right.
Absolutely, yeah.
Wonderful. And so now with that, I'll hand over to my colleague and fellow volunteer, Danielle, who will take us through our fun little poll.
Great, thanks, Nikki. So we thought we would do a little bit of a dilemma with you. And so we've termed this our desert island dilemma. And so after an unfortunate and series of events, you find yourself stranded on a desert island in a tropical area, and rescue could be just around the corner. And so this is a test in decision making. We're going to give you a few options and then put up a poll for you to make your decision, and then we'll see what that says about you in our sort of pseudoscience pop quiz. So here are the options. So you're allowed to pick 2 from the following objects to help you on your desert island. So your options are fruit and vegetable seeds, a Swiss Army knife, fishing net, sunblock, a 100-foot rope, a waterproof sheet, a metal bucket, a lighter, or insect repellent. So what we're going to do is, if you can either scan this QR code with your phone and it should take you to a website, or you can head to slido.com and enter the code 465639. You should see this question and see the options and be able to select 2 options. So what we'll do is we'll leave this slide up for you for about 5 minutes. We'll have a little bit of a break now, and then we'll come back to this at the end of the second talk and see what options everybody chose. So just have a look and see if we've got any options rolling in. So I'm starting to see some results, so that's great. So if anyone else would like to join in, we'll leave this slide up for you, and we'll just take a quick 5-minute break now, and then we'll head back towards you. Thanks.
Hi, everyone. We're just going to cut the break a little bit short so we can stick to time and go through some of the answers at the end. Don't want to cut any of it short, though. And so now we're going to introduce our next speaker, Dr. Armin Lak, who's a Wellcome Trust Sir Henry Dale Fellow at the Department of Physiology, Anatomy and Genetics. And he's going to be talking about confidence versus reward. How does your brain learn to make the best decision? Given that we are, bombarded by so much at the moment, making the right decision might be a bit difficult. So maybe Dr. Armin Lak will be able to help us with that. Thank you, Dr. Armin.
Good evening, everybody, and thank you very much for joining us this evening. My name is Armin, and my lab at Oxford is studying neural base of learning and decision-making. We are in Pint of Science this evening in the virtual format, but let's imagine that we are in non-virtual version of Pint of Science. You're getting to the pub is crowded, and you're trying to choose a drink before the talk is starting. So you're looking at this scene. There are many things that you can read. You can remember which beer that you like most the last time that you were in this pub. You might think about the prices of this beer. You might smell some of the beers more than the other. You might think about whether your drink want to be cold today or not, and you make decision by considering all these options together. All of them are coming together. The color of the drink, the smell of the drink. Is it cold enough? Is it too expensive or not? Do I like it or not? And all this information is really coming into the brain. They are represented in the brain. Some of these, like color, smell, or the temperature of the drink, are coming from outside. We are feeling them with our senses. They are external information which are coming to the brain. And the others are in fact internal to the brain. They are information that we have from our past experiences. Like we can remember how much usually this pint of drink is. Or we can remember how much we liked this drink last time. And making choice is effectively the process of combining all this information together and coming up with a choice that we like most. To do so, the way that behavioral economists or psychologists would think about the problem is that we are considering all this type of information, and we are trying to generate a single number for each of those drinks. What is the value of that drink? How much I like that drink? And we can then use this value information in order to make choices between options. So you have the value of the first drink, we have the value of the second drink, and the idea here is to compare values and make a choice between those. With every of this decision, also coming a sense of confidence. So when making the choice, we always carry that sense of confidence. And by this, I don't mean anything such as self-esteem or self-confidence. What I really mean is a simple sense which tells me how sure I am that my choice will be correct. How sure I am that the values that I have computed are correct. And this confidence signal itself is playing an important role in the process of decision making. So if I have choices and I have some value estimation for those, if I do have confidence about these choices, which is very different, then this might really influence my decision making. I might have an option which is very valuable, but I'm less confident about that option. And I might have another option which is less valuable, in this case on the left side, but I'm very confident that I would get that reward. And this confidence signal might even change those choices that we make. We eventually might go with the one which is lower value, but we are very confident about it. So I'm saying all this, showing all this to effectively convince ourselves that decisions that we are making are informed by different signals, and two most important of them are value of option and how confident we are that we have computed those values are correctly and we get the reward that we expect. Okay, so the question that my lab is trying to address is how the brain is combining this information for making decisions. as Kerry was telling us, the brain consists of neurons, billions of neurons in human brain, and they are making trillions and trillions of connections between them. So the question for us is how those individual neurons embedded in the tissue of the brain are working together. What are they signaling during this type of decision making? Can they combine value information and confidence information in order to guide choice? Okay, so the other question even behind it, before this question, is how we can approach this problem. If we want to do that, we need two things. We need to be able to measure decision making in a controlled fashion. We need to be able to define specific parameters in the decision process and control those in the context of experiment and observe the choices that the subject is making. And also we need tools which can allow us to measure or manipulate the activity of individual neurons. And we do have those tools in animal models, just like Kerry was nicely explaining. There are tools that we have in animals, and they are allowing us to record from each of those neurons, or even change the activity of those neurons during the task. So the situation for us is to bring these two together, to have these tools, and also to define behavioral experiment, in this case in animal, which can allow us to think about this problem of decision making in the laboratory context. So how do we do that? We use mice, and we are introducing mice in very simple experimental decision making tasks. So animal is looking at this monitor, and it would need to decide whether this stimulus which is on the monitor is on the right side or on the left side. So if the stimulus is on the left side, an animal would press the lever on the left, it would get a reward, and it would press the lever on the right, it would get no reward. And in the other condition, other trial of the experiment, the stimulus is on the other side. If the animal this time pressing the lever on the left, it would get no reward. There's no stimulus on the left. And if it presses the lever on the right, it will get reward. Okay, so that's the basic of the behavioral task. But what we really want to do is to manipulate or change the confidence in the task and value of those decisions in a systematic manner. Okay, so how can we go about changing confidence? So let's think about the situation that we have those stimuli. And one way to change those confidence is to change the contrast of the stimulus. So this time I have a stimulus which is less bright. So that's probably the case that animal would not be able to see them sometimes and make choices which are low confidence. And other situations, we are playing with the position of the stimulus, we are changing the contrast of the stimulus, and we're creating situations that animal is going left and right, making choices, but the stimuli which are coming in for that trial are either very bright, very easy, very high confidence trials, or lower confidence and the lower brightness or contrast that you can see in those examples. That's one way to change the confidence. And we would also like to change the value of reward. So to do so, we are now introducing the reward, which are different sizes, like the cheese, the small cheese, or large cheese. And in that trial 1 up there, if the mouse reports, because the stimulus is on the left side, if the mouse is reporting left, it will get a small cheese. If the mouse is reporting right, it will get no cheese because there is no stimulus on that side. And if we go, for example, to the third trial is an excellent trial. The stimulus is very bright. Animal can very confidently see that stimulus, and the reward that animal would get for that choice is very big. So these mice can perform this task, trials after trials. They like those rewards, and they do hundreds of trials per day. And we can count the number of times that animal is going left or right for each of those stimuli and each of those rewards. And we can make curves which are looking like this. So we have the contrast of the stimulus on the x-axis. It's either the stimulus on the right side or on the left side. And we are counting how many times an animal is making right choice or left choice. And we get curves like this from mice. So they are doing quite good in reporting the position of the stimulus. And if we change the size of those rewards, this curve might shift to the left or to the right, depending on which size of reward is where. Okay, so having this behavioral paradigm, now we can use the tools that we have in neuroscience to ask the question of whether neurons in the brain can combine value and decision confidence. For tonight, I'm going to only focus on one specific class of neurons in the brain, the dopamine neurons. And those are neurons which you might have heard about them in the context of Parkinson's disease, as well as other psychiatric disorders. But they also have very important role in processing reward in the brain and learning based on reward properties. So these neurons are deep in the brain. You can see on the right side in the human brain in this cartoon. And when they are active, they are releasing this dopamine, which is a molecule, in the rest of the brain, and they influence the rest of the brain. They change the way that brain is working in those situations. We have very similar anatomy in mouse brain. You can see in this figure on the left, we are marking now dopamine neurons, and those neurons, just like human neurons, they are having branches which go into the rest of the brain, and they're influencing the neural activity, the brain activity in the rest of the brain. So having these dopamine neurons, what we know about them is that they are very much active and they are very much responding to rewards. So if our mouse is pressing levers and it would get a large piece of cheese, then dopamine activity would go up. And if the reward is small, then dopamine activity would go down. And the question that we want to ask is that, given what we know in this reward processing, whether this neuron can combine value information and confidence information during decision making. So to do so, we can do a variety of things. Either we can use electrophysiology, like the way that that Kerry was describing, looking at action potential of those neurons, or we can use another tool which is based on genetic and light. So this way, using some genetic tricks and optical tools, we can measure the activity of dopamine neurons using light. So we're shining laser on those neurons, and when they are active, they are shining light back. And we can collect those lights that neurons are sending to us, and we can visualize that. And we see something like this. Over time, the signal is changing, going up and down, and it is happening at different times during the task. So this is effectively giving us a precise readout of dopamine activity during this behavior. So we are now going to look at those activities in the context of that decision-making behavior which we just talked about. And we have our figure, we have our curve, but this time on the y-axis, on the vertical axis, rather than we have dopamine. We have the level of dopamine in each of those conditions. And if we plot dopamine activity in this condition, what we can observe is that these dopamine signals are very strongly changing based on the difficulty of the stimulus, based on how visible the stimulus is. So regardless of whether the stimulus is on the left side of the screen or on the right side of the screen, as long as the contrast of the stimulus is high, and as long as the choice is easy for the animal, those signals are growing. Now we can bring in the value component, the differences of the sizes of cheese. And what we can observe in this situation is that if we now look at those specific situations that the larger reward was on the right side in this case, then that dopamine curve is shifted. So it's growing on the side which has larger reward compared to the side which has a smaller reward. And from this form of analysis that I'm showing here, we can conclude that these neurons are really coding two different things. They are signaling two different things. They care about how valuable the reward is. They also care about how much of those, how confident the animal is in receiving those rewards. Here we are parameterizing that confidence by changing the contrast of the stimulus on the screen. So these neurons are in the position to combine these two types of information. And these are very distinct type of information. If you recall, in the beginning, I was talking about external information and internal information. So that reward value, this is something that is internal to the animal. He has learned it over time. And the stimuli, these visual stimuli on the screen, are an example of external information. They are just coming on that trial, on that moment to the animal. And these neurons are managing to combine this type of information and signal a quantity which is combining these two very distinct forms of information. So this is really establishing the correlation between the activity, in this case in dopamine neurons, and the behavior of the animal in every moment in time during the task. We can even use other forms of technologies in order to influence the activity of these neurons. So we use other forms of genetic tools and genetic tricks, and this time we would use light in order to activate or inactivate these neurons. So we shine light in order to activate or inactivate these neurons. And this can be very specific. For example, we can shine light for a very brief duration of when the animal is seeing the stimulus. Or we can do so, again, very briefly at the time that animal is receiving those cheese reward. And we can then see how this brief manipulation, this brief activation of dopamine activity influencing the behavior. I'm not gonna make complicated graphs about how this is going to happen, but this tool is allowing us to really go beyond the correlation and move toward studying causation, move toward understanding of whether these signals are actually playing a causal role in changing the behavior or defining the behavior of the animal when it's making decisions in this laboratory context. With this, I'm going to leave you with few things. And my whole point here being that when we are in the pub, when we are making simple decisions, even these simple decisions are very, very complex decisions. As I said in the beginning, they require integrating information from our senses and putting them with what we have in mind about the value of each of those options. And in neuroscience, we have now tools which is allowing us to study these decisions. We translate this complex decision into a situation that we can study in the lab, and we use those tools in order to manipulate or measure the activity of neurons in the brain and understand the relationship between neural activity and behavior. But we are really in the beginning of this journey. As I said, there are billions of neurons. I only focus on subpopulation of these neurons, in this case dopamine neurons. We are having just the start of what is going to be a very interesting decades of research in understanding the brain signal underlying behavior. I showed you examples in dopamine case. These neurons are really smart. They care about the reward value. They also care about how confident the animal is in getting those rewards. And this is really putting these neurons in a very unique situation of combining this information. Perhaps they have a very critical role in bringing together various type of information and guiding the behavior on a moment-by-moment basis. With this, I would like to thank people in my lab, the funding which allow us to perform this research, and thank you very much for listening. And I'm happy to take questions.
Thank you so much, Armin, for a really sort of insightful talk. And with that, I think we have time for one question, maybe two. I'll start with the first one from Claire. Do you find confidence in decision-making to be reduced in Parkinson's disease?
That is a great question. There are not, unfortunately, that many studies yet on this matter. This is something that I would say we would need to do, and a lot of neuroscientists which are working in the interface of clinical neuroscience and basic neuroscience would be in the position to address these questions. But at the moment, there is no clear answer yet to that question.
All right, thank you. I think because we're short of time, I'm going to hand over to Danielle to discuss our poll results. But those of you that still have burning questions, maybe you could tweet our speakers, tag Pint of Science and hashtag Pint21. 19? I'm 2 years behind. Pint last year didn't happen. And maybe they'll be able to answer your questions offline here. Thank you so much to our speakers. And now over to Danielle.
Thank you to everyone who entered the poll. We had over 80 responses, which was a really great involvement from everyone that's watching. So the results are in, and I can tell you that the most popular answer was knife, well, Swiss Army knife, followed closely by the waterproof sheet. And our sunblock and repellent and seeds and rope were sort of least desirable options. And so thinking about what you've picked, we've put together a little bit of an idea about how we make our decisions. And so there are 4 types of personality that will feed into how you would make these decisions in this sort of scenario. And so depending on what you picked, you might be able to fit yourself into one of these 4 boxes. And so the first, if you picked the rope and the bucket, would make you a synthesist, and they're defined by creative and curious nature, and instead of leading with logic, they love to explore more abstract ideas. Our idealists might have chosen the seeds and the fishing nets because they're future orientated and they are thinking about what's going to happen in the future. Our pragmatists, who we seem to have a lot of this evening, chose the knife and the waterproof sheet, or perhaps the lighter. And they don't waste any time, they just like to take action, while our realists are dealing with the real world situations and might be more concerned about how their welfare is in this time. So they would be interested in getting the sunblock and the insect repellent. So this was just a bit of a game for you guys. And as a disclaimer, I will say this is for fun. Please don't quote me on any of these choices. But I hope that it just gives you an idea of the decisions that we make and how they can be defined by the type of choices that you make. And today we've heard a little bit about the science of that. So I'll pass you back to Nikki to finish up. But thank you again for everybody who joined in with our poll this evening.
Thanks, Danielle. And thank you to all of you for joining our broadcast this evening. I know it's not the same as being in a pub, but, you know, this really kind of helps push the boundaries forward of kind of scientific outreach. And again, I'd like to thank our speakers, Dr. Armin Lak and Dr. Kerry Walker, for their time this evening. If you have any more questions, please feel free to tweet them, tweet us, use the hashtag Pint21, and also tag Pint of Science in your questions. And with that, I'll draw a close to this evening's festivities, and hopefully we'll see you again soon. Thank you and goodbye.